Okay, I left off a little bit differently in each class. So in all classes, I talked about this, the leading and the lagging strand. And most classes I left off with this. I drew this on the board. And I had here the 3' and the 5' end. And if this is the replication bubble, I asked you which would be the leading strand. And this down here would be the leading, and this would be the lagging strand during replication. And let me explain why that is. So here, if this is the 5' end, if you're going to make the complementary strand, then that means that this has to be the 3'. And if this is going to be the leading strand, this is the 3', the new complementary strand, this would have to be the 5'. And so what happens, though, is that then this is leading because the new strand in green is going to grow from the 5' to 3' direction. Every new strand needs to grow in that 5' to 3' direction. In this case here, this is the lagging. To grow in the 5 to 3' direction, it has to start here and go to the end, then restart and keep restarting, and so that's why that's the lagging. But the thing is that DNA polymerase cannot, and we're going to write here, and I did this in some classes, initiate synthesis of a polynucleotide. They can only add nucleotides to the 3' end of an existing chain of nucleotides. So then what happens is the initial nucleotide strand, the very beginning, is a short one called a DNA or RNA primer. For a lot of our genes, it's an RNA primer. And so an enzyme called primase can start the RNA chain from scratch. So therefore primase, see what DNA polymerase can't do is it can't come to this and start adding nucleotides. It can only add nucleotides if nucleotides are present. But when you first start the strand, the very beginning, there are no nucleotides present, and so therefore DNA polymerase can't start replication. There's no nucleotides here, so it can't start growing it in the 5 to 3' direction. So therefore replication cannot occur if we depend on DNA polymerase by itself. So what happens is this enzyme called primase is unique in that it can add nucleotides to a spot where there's no nucleotides present. So what happens is right here, the 5' end, this is your RNA primer that primase adds. And then once that primer is on there, then DNA polymerase, now there's existing nucleotides. And so therefore DNA polymerase can add to that and begin to grow the strand in that direction, in the 5 to 3' direction. And that is the leading strand because it'll go to the end as keeps on unzipping and so on. Now if we look at the lagging strand, we need a primer. Let's say it opened up this much to begin with. So it adds down here it adds the primer here. So here it needs to add the primer right here to begin the replication, and then DNA polymerase can go add nucleotides to the end because it's growing in the 5 to 3' direction. 5 to 3' direction. So it gets to the end, and now the DNA unzips some more, and yet we need another primer. So every single time it unzips, and on the lagging strand, replication starts all over again from the 5 to 3' direction. So we add a new primer, and so there has to be a new primer on every single Okazaki fragment. All right, and so therefore the lagging strand has lots of primers. The leading strand has just a few. So therefore for this bullet here, only one primer is needed to synthesize the leading strand, but for the lagging strand, each, we're going to write Okazaki fragment must be primed separately. All right, and so that's what this picture illustrates. All right, and that's what the next picture illustrates as well. In a little bit different way than mine. Okay, so they're showing you just one side of the DNA strand over here. So they're showing you the it's going to grow in the 5 to 3 prime direction. So this is the lagging strand. So this is looking at just one side. So primase is adding the RNA nucleotides. And so it's adding the primer in the 5 to 3 prime direction. So now once the RNA primer is there, then in DNA polymerase 3, there's different DNA polymerases. DNA polymerase 3 begins adding the nucleotides to the end. So this is the lagging strand, because notice here it gets to the end and DNA polymerase comes off. So this right here, this is all about the lagging strand. So then now we have to start over here if it hasn't unzipped, and now we're going to unzip it some more. And so now we need, it doesn't show you here. Here's the something I don't like about this is that when it starts over here, you have to make a new primer. So I'm going to put here, this is the RNA primer. ER primer. And then, so once the RNA primer is put on here by primase, then the DNA polymerase continues adding nucleotides till it gets to the other Okazaki fragment, all right, and then it'll come off, and this will keep on happening. Then upstream here, you'll have another primer, it'll add nucleotides. But the thing is eventually, we want two things to happen. We don't want our DNA embedded with all these RNA nucleotides. So, like if we look back at this picture I drew before, this one, we have all these RNA nucleotides interspersed in our DNA, and we want all DNA. Remember, we're copying DNA. We want it to be all DNA. So therefore, we have an enzyme called DNA polymerase 1. So DNA polymerase 1, we'll write about this here, replaces the RNA nucleotides with DNA nucleotides. So it switches them out. And then in comes ligase to glue them together. So at the very end, you get a full replicated piece of DNA. So going back to this picture here, what it would look like after DNA polymerase 1, DNA polymerase 1 takes the RNA nucleotides and replaces them. So all the orange ones would be replaced with green. And then ligase comes and glues them all together so that they're no longer in fragments, but in one continuous nucleotide chain. So let's write about these other proteins. So a couple of things so that's the big picture as far as what happens. And so this enzyme called helicase, way at the beginning, to untwist the DNA, helicase untwists the double helix and separates the template DNA strand at the replication fork. Then you have these what are called single-strand binding proteins binding to and stabilizing the single-stranded DNA until it can be used as a template. So remember, you have your DNA molecule, so we have to untwist it at the beginning and separate the two strands. But what happens is that those two strands, we don't want them to go back together. We want to keep them apart so that we can add the nucleotides. So that's what those single-strand binding proteins do, is they keep them from rehydrogen bonding back together. Topoisomerase, it says correct overwinding. This is going to be not on our test, so not on test. You don't need to know that one. All right, primase synthesizes an RNA primer, so we talked about that at the beginning. All right, at the 5' end, so the thing can grow in the 5 to 3' direction. Then what enzyme is the one that adds the nucleotides continuously to the leading strand and elongates Okazaki fragments? That is DNA polymerase. And then there were numbers. Do you remember which one it was? 3 or 1? If you said 3, you would be correct. All right, so then the one that removes the primer and replaces it with DNA is DNA polymerase. And you probably, by process of elimination, should have guessed 1. And then what glues them all together, joins the DNA strands to hook them all together? Do you remember? It is DNA ligase. Joins them. Okay? And so those are your major kind of players in the role of DNA replication. So this picture here gives you an overall view as far as what's going on here. So if we look here and you look at what's going on, you have your leading strand, you have your lagging strand, and you've got all kinds of things, everything going on at the same time. What thing happens first? See if you can think about what happens first. If you need to pause this, check yourself to see what you would say on this happens first. After you paused it, you should have come up with that the helicase has to unwind and unzip or break apart the double helix. And so on this picture, which is helicase? If you need to pause it, do so. Helicase is right here. All right, so I'm going to write here. So this is step number 1. Helicase unwinds and unzips. That means to break apart the hydrogen bonds, the double helix. That's step 1. So that's your helicase. Now look at this picture and think about what happens next and what proteins are involved next. Pause it if you need to. What happens next is these guys here, the single strand binding proteins. So these guys right here, put over here. So these are step 2, single. And this, I think, is where I left off with first hour. Everybody else didn't get to this. Single strand binding proteins stabilize the unwound DNA. What does that mean? It means that it keeps it from hydrogen bonding back together. So then what I have for step 3 and 4 is about the leading and the lagging strand. I'm going to make the leading strand step 3, but really those two things happen simultaneously. So over here with the leading strand, we're going to put here step 3. This is made continuously in the 5' to 3' direction. And so in this case here, how do we know it's the 5' end? Well, let's look at over here. This is the 5' end of the original dark DNA here. That means that has to be the 3', which means the new strand, the complementary strand we're making, this is its 5' end, so it's going from 5 to 3'. All right, so that's what's going on there, and that's how we know it's the leading strand. And so this would be over here the 3' end of that strand. All right, so at the same time that's happening here, for the lagging strand, this lagging strand, that's step 3 with the leading. The lagging strand, let's put step 4. This is not made continuously, it's made discontinuously. That's right, primase synthesizes a short RNA primer, which is extended by DNA polymerase 3 to form an Okazaki fragment. And that's what we have here. So we've got, here's the 3' end, so that means this side's going to be the 5' end of the new strand. So it's 3', so this is going to be the 5'. And so this is growing in the 5 to 3' direction. So remember, each time as it unzips, we make a new fragment. And this is beginning the new Okazaki fragment here. Notice that there's a primer that was added. And so over here, they're showing you primase on the next fragment. So primase is making the primer right now for the next fragment. It's already done here. The RNA primer is already done, and DNA polymerase 3 is now adding the nucleotides till it gets to the beginning of the next Okazaki fragment. After this primer is built, DNA polymerase will come over here and build. All right, so what happens to those Okazaki fragments up here? So we have DNA polymerase 1, remember what that's doing? It's replacing, so this is replaces RNA primer with DNA. So it's doing that. Notice up here with this fragment, our primer is already replaced. So DNA polymerase already did that here, and now we have ligase. Remember, that's the gluing together, glues fragments together. So this is going on at the same time new Okazaki fragments are occurring right here. All right, and glues them together. And so one last thing I want to write, let's write it over here about those things. So we'll call it step 5. After the RNA primer is replaced by DNA polymerase 1, DNA ligase joins the Okazaki fragment to the growing strand, so that you end up with, after ligase is going to glue all these pieces together, you have your strand growing, and it's growing in this direction ultimately, even though it's being built left to right. And that is the role, or all the parts that go with DNA replication. Alrighty. So then what happens, doesn't happen very often, but what happens if there's a mistake? So this is proofreading and repairing DNA. Luckily, if the mistake gets added, let's say the DNA polymerase tries to add a T to a C, and you know that T is supposed to go with A. So we have enzymes, other classes of DNA polymerases. We talked about 3 and 1. There are different classes of them. Proofread newly made DNA, replacing any incorrect nucleotides. So in mismatch repair of DNA, repair enzymes correct errors in base pairing. So they correct the errors and replace them with the right base, and that's called mismatch, where they're matched wrong. Then there's nucleotide excision. This is where we have to cut out some nucleotides. In this type of repair, enzymes cut out and replace damaged stretches of DNA. So we go to this picture here. This is a common situation in which this excision repair would work. Notice here it says a thymine dimer distorts the DNA molecule. So these are T's, and it hasn't necessarily made a mismatch. It still is with an A, but if you notice here, do you see how these T's are bonded together this way? There's that little line right there that bonds them together. That's not supposed to be there. And that binding that way, instead of, they're supposed to be hydrogen bonding with the bases across from it, distorts and changes the shape of, and you see this, of the DNA, and you see this bulging. And so that's what a thymine dimer, that's what it means by distorting the DNA molecule. So an example of something that could do that is UV damage, ultraviolet light. So in comes an enzyme called nuclease. So this I'm going to have you box in. This you need to know. It cuts, you need to know this one. Nuclease cuts the damaged DNA at 2 points. So here's the cut right here. That's what this arrow is, and a cut right here. So 2 cuts on either side of the damaged DNA and removes that damaged section. So then we have to replace that damaged section. So in comes another DNA polymerase that repairs it and adds the correct nucleotides, fills in the missing nucleotides, and then we have to glue it together. What glues it together? DNA ligase. And now you have a fixed, quote unquote, fixed DNA molecule. All right, and so no longer is damaged anymore, and so that's called excision, nucleotide excision repair. So we're cutting it out. Otherwise, the mismatch repair, just if it accidentally puts the wrong nucleotide, takes it out and puts the new nucleotide in. So luckily we have enzymes that catch most of our damaged DNA, most of our mistakes and so on, and so we rarely see repercussions from that. So in here it says, though, that this imperfect nature of DNA replication, because there are mistakes that can happen, and sometimes they aren't caught, the imperfect nature of DNA replication and repair can increase variation. How in the world could it increase variation? Well, if a mistake occurs in your DNA, and then that mistake allows individuals to produce a different protein, and that protein gives a trait that is beneficial, then that introduces a new trait or new gene into the population and therefore increases variation. And so remember, that's the stem of how all different types of genes originated to begin with. Why flies have red-eyed and white-eyed genes. Remember, the white-eyed was the mutation, the red-eyed is the wild type. And so how do we get those different colored eyes? The original gene was mutated, and therefore increasing genetic variation. And the last little part of this is about what happens at the end of DNA molecules. So back over here, I should go back to this. When we talked about how you need a primer and how the primers are replaced with DNA nucleotides, the problem is at the ends of the DNA, because DNA polymerase that replaces the primers with DNA nucleotides still can't add to the end of a growing chain. So let me explain what this means. It says limitations of DNA polymerase create problems for the linear DNA of eukaryotic chromosomes. The usual replication machinery provides no way to complete the 5' ends, so repeated rounds of replication produce shorter DNA molecules. Eukaryotic chromosomal DNA molecules have at their end nucleotide sequences called telomeres. These do not prevent the shortening of DNA molecules, but they do postpone the erosion of genes near the ends of DNA molecules. So let me explain what that means. Okay, so we're going to draw a series of pictures over here. So here's a kind of similar to the picture we drew before. And so here's the 3' end, here's the 5' end. So when we make our new DNA molecule, this needs to be the 3' or the complementary for each side. And so what happens is, remember that we have one primer here and we have multiple primers here, where this one primer here will build the strand continuously, and that's why that's the leading strand. This will build to the end, but then need an additional primer to build the next Okazaki fragment, and so on. So with that being said, what happens is that when we finally get to the end, let me draw this here. So this is the same molecule. Here's the 5', 3', 5', 3'. And then here, we're going to have the complementary strand, this is going to be the 3 and the 5 prime, 5 prime, and 3 prime. So what happens is, remember at the original, you had one primer, and then this continues on. Here you had multiple primers, primer and built in this direction, primer and built in this direction. I'll just do 3 here, primer and build in this direction. So you got these fragments, and so you have a whole bunch of primers to replace, because remember they're RNA, we want to replace them with DNA, and here you have one. All right, so what happens is that, let's get a different color here. So all these primers, the mRNA primers, are replaced with DNA, right? So I'm going to go ahead here. So and then ligase hooks them together. So what happens, if we look over time here, so here's this, 5 prime, 3 prime, 3 prime, 5 prime. The new one here is 3 prime. It's going to be 5 prime, 3 prime. This is 5 prime. All right, so when we draw this, so now pretend like this. All right, so DNA polymerase is going to come and replace this orange with green. So it's no longer going to be green. And then ligase is going to hook them together. So what's going to happen is that the line is going to be all green. The problem, though, is at the ends here, these two ends, when you take away, pretend like we take away these orange ends. So we take them off, but the DNA polymerase that replaced these guys, there's no existing nucleotides here, so I can't add DNA prime nucleotides. That's the whole reason why we needed a primer to begin with, because even on the leading strand here, DNA polymerase can't start with nothing there and start adding nucleotides. So therefore, that was the purpose of adding a primer, so it can add the nucleotides and so on. Well, what happens is now that you've added the nucleotides and you take away that primer, DNA polymerase can't replace that primer, and so that happens only on the ends. And so what that looks like is this. So you're not going to have, wherever that orange at the end was, that primer, it disappears. We take it off, but DNA polymerase can't replace it. So therefore, this looks like this, where this part's not replaced. The end here isn't replaced. And this one, we take the primer off, but we can't replace it, so it starts this way. So therefore, here we have right here and right here, we have missing nucleotides at the 5 prime ends. Now, that's a problem. So then imagine the next time our cells divide and we have to replace this DNA, and each one of these DNAs replicate, then we're going to have even shorter 5 prime end, and even shorter. Every time it replicates, that end gets a little shorter and a little shorter. And so that's a problem, because what's the purpose of our DNA? To make proteins. It has genes. So therefore, we have some protection measurements, and that's what we wrote here about these telomeres, that there are these nucleotide sequences that don't code for making proteins at the very ends of our chromosomes, and so the ends of our DNA. So therefore, as we replicate our DNA and the ends get shorter and shorter and shorter, for a long time, it doesn't matter that they're getting shorter, because the DNA that we're missing are not actually coding for genes. And so therefore, it doesn't hurt us for a long time. All right, so that's what it means, that the telomeres do not prevent the shortening of DNA. The shortening does happen, but they do postpone the erosion of genes near the end of the DNA molecule, because they're not genes, the telomeres. All right, and so that's a protection measurement. Eventually, the more times a cell divides, eventually, like if this is let's do here, like if this is, let's say, the start of genes here, and right here, eventually over time, as the DNA keeps replicating and the telomeres get shorter and shorter, it'll eventually start getting into the genes and therefore affecting us. But it's not till later on. And I think that the shortening of the telomeres here and the ends of the chromosomes have to do with the aging process and what happens as cells get older, because those older cells have divided more. Okay. So the problem becomes, and what these last two bullets are, is making egg and sperm cells. When we make egg and sperm cells, we want to give the full DNA molecule, one of every kind, to our kids in our egg and our sperm cells. So the babies have all their DNA and they don't have shortened chromosomes. So that's where this bullet says if chromosomes of germ cells, now what in the world is a germ cell? Germ cells, let's put over here, germ cells divide to make gametes. All right, so how do we make gametes in meiosis? So remember there are 2 cell divisions in meiosis, 1 to 2 cells and 2 to 4 cells. This whole process is meiosis. These guys right here are your gametes. This right here is your germ cell. So germ cells are in testes and ovaries to divide to make gametes. So if chromosomes of germ cells became shorter in every cell cycle, essential genes would eventually be missing from the gametes they produce. That's no good. All right, we want to get full pieces of DNA to our kids. So an enzyme called telomerase catalyzes the lengthening of telomeres in germ cells. So we have this special enzyme, remember ACE is the enzyme, special enzyme that just in our germ cells takes any shortening, and telomerase can add nucleotides to the end and fix it. So telomerase, if I were to act as telomerase here, telomerase can go over here and fix and add the missing part here and add the missing part here so that our germ cells have the correct amount of DNA and we don't have babies that have shortened chromosomes and therefore are kind of behaving as older cells than they actually are. So this telomerase only found in germ cells. Only in germ cells. So you can imagine that there's a lot of research being done with telomerase and how can we prevent the shortening of the telomeres and this whole multi-billion-dollar industry of anti-aging. It's a big deal, all right? And so there's a lot of research being done with that. And that is the end of Chapter 16. So I hope you guys enjoy the rest of your break, and I'll see you on Wednesday.